can't advance my slides. Okay, perfect. So I'm putting up this table, which might not contain everything, but just to give you kind of an overview of the classical subdivision of vascular anomalies into vascular tumors and malformations, and highlighted are some of the genes that my lab has been working on. So just to kind of give you a broad overview. But today I'm going to be mostly focusing on PIK3CA and Tie2 or TEC. And as Mike mentioned, so we have been working on PIK3CA in the context, a little bit in the context of venous malformation and in the context of capillary lymphatic venous malformation or CLVM or CLOVEs. So just going into that, CLVM is called capillary lymphatic venous malformation, and this is found in patients with CLOVEs and is characterized by an aberrant network of capillaries, lymphatic vessels, and veins. And also in many patients, there is soft tissue overgrowth. These lesions are congenital, and so they actually start to grow during during the fetal development, and they enlarge with time. And as Michael mentioned, though most of these patients have PIK3CA mutations. Today, so we have actually a set of targeted inhibitors. Among those, the P110 alpha specific inhibitor BYL719 or alpelisib, which has been FDA approved very recently, and also AKT and mTOR inhibitors such as rapamycin are in clinical trials and have a good degree of efficacy. So you probably all know this very well. I'm just going to very briefly. So the PI3 kinase is composed by a regulatory and catalytic subunit. P85 is the regulatory, and P110 alpha, which is the subunit specifically targeted by alpelisib, the PI3 kinase inhibitor, is the catalytic subunit. Mutations happen specifically in the catalytic subunit and induce constitutive activation of the pathway downstream of PI3 kinase, leading to increased AKT and mTOR signaling, which leads to increased cell survival, proliferation, metabolism, and angiogenesis. It is of interest that PIK3CA mutations are not only found in vascular anomalies, but are also found in cancer. And I want to just point out to you that the PIK3CA is the second most mutated gene in cancer. And bringing back to vascular anomalies, the same hotspot mutations found in vascular anomalies at the site C420, 542, 545, and 1047 are the same found in cancer. Now, what is different about vascular anomalies and cancer, we still don't know enough, but I wanted to bring up a nice paper or article published by Robert Semple and Ralitza Madsen, where they've specifically tried to address this, to answer this question. And what they are showing here is that in cancer, the PIK3CA mutation happens in a form that is almost like if it was a biallelic mutation or a homozygous. So either there are two copies of the mutant allele, or there is a mutation in a gene in the PI3 kinase pathway that is also inducing constitutive activation. So which results functionally sort of as if it was a homozygous hyperactive mutation. Conversely, as you know, in vascular anomalies, this mutation happens in heterozygosis. Only one allele is mutated. And what they've done to show the differences between a heterozygous and homozygous mutation, they have taken a biopsy, skin cells, and reprogrammed them to become induced pluripotent stem cells. And then they CRISPR in the mutation as a heterozygous or homozygous. And they have looked at the differentiation of the induced pluripotent stem cells into the different lineages, and also they have done they have looked for differentially regulated genes, upregulated and downregulated, and they performed the two following comparisons: homozygous versus wild type and heterozygous versus wild type. And what they've seen is that when you compare homozygous mutation versus wild type, you have many more changes, 977 and 911, up or down, compared to when you compared to the comparison heterozygous versus wild type. And the genes that seem to be importantly upregulated are genes that promote stemness of the pluripotent stem cells in the homozygous. So they think that in cancer, this mutation, the PIK3CA mutation in homozygosity, promotes stemness of the cells. So here's the question, the mutation, homozygous, heterozygous, and another important question of the field, which I think we all talk about it these days, is when is the mutation happening, and is there a difference if the mutation happens early versus late during development? And this publication from Taija Mäkinen has very elegantly shown that if you overexpress the PIK3CA mutant allele driven in VEGF receptor 3 expressing cells in lymphatic endothelial cells, and you induce expression of this mutation at 2 different time points. First is during early embryonic development, E11, or after birth at P2, you get a completely different phenotype. So when you do early tamoxifen induction, you obtain macrocystic lymphatic malformation. And when you do it later, there is formation of microcystic. And this is another way of showing this. This is the skin ear, and this is a control. This is a mutant, early embryonic, and you see this enlarged lymphatic channel, macrocystic lesion. And here you can see later induction, a microcystic. And so what we were interested in addressing is because CLVM is formed by capillary, lymphatic, and veins, is in which cell type is the mutation happening. And so we were just trying to think, okay, so PIK3CA mutations are found in CLVM, but they have also been found in lymphatic, in isolated lymphatic malformations. This is some of my work, but other people have shown this. And in venous malformation, isolated venous malformations, also PIK3CA mutations have been identified. Conversely, in capillary malformations, we have mutations in GNAQ. So which one is the cell type of origin? And to address this question, Gillian Goins in my lab has devised this protocol to isolate endothelial cells from excised tissue from patients or sclerotherapy, blood or lymphatic fluid. And the cells get plated, and then we do a CD31 immunomagnetic isolation, and we pull down endothelial cells, and we separated them from the non-endothelial, and then we investigated for presence of the PIK3CA mutation. First of all, we found that the mutation was exclusively found in the endothelial cell population and was not found in the non-endothelial. And with these endothelial cells from patients, we generated xenografts by expanding the cells in vitro. And then these cells are injected into immunocompromised nude mice, resuspended in matrigel, which is an extracellular matrix that is fluid at 4 degrees, but solidifies at 37 degrees. So keeps the cells in the place where we inject them. And we don't add any other factor. We know matrigel has some factors, but we don't add any other. And as you can see here, so those are sections of the lesions that the cells formed of the xenograft. And the cells from patients form vascular lesions that are similar to the histology of the patients from where we obtain the cells. You can see here, this is the identifier. We call this patient A. This is a xenograft, and here we have the patient histology. Here we have another patient F, xenograft, and histology. But really, at this point, the question that we had was, again, which one is the cell type? And we know there are different subtypes of endothelial cells. And so we have lymphatic, vein, and capillary. So the capillary part, we know there are 2 reasons why we can't exactly look into that. We don't have specific markers to look at these cells in vitro. And also the capillary stain usually in CLVM patients tend to be superficial. So we never get really that part in the sclerotherapy or in the resection. So we try to focus on at least dissecting down if the mutation was in the lymphatic ECs or vascular ECs. And to do that, we needed to generate clones derived from one single cell. And so work done in collaboration with Tim Lacrosse, and beautiful work done by Trisha, and then in his lab, and Jillian and Nora in my lab, have performed a limiting dilution to seed one cell. The plan is that you will seed one cell every 5 wells. And then we specifically expand the populations that, that can expand. And so as you can see in this Western blot, we were able to get several clones. You can see 1, 2, 3, 1, 2, 3. And the way we know if or vascular is, okay, first of all, let me go down to PROX1. So PROX1 is expressed only in lymphatic ECs. And you can see in the 3 clones of LECs, it's expressed, but it's not expressed in the vascular endothelial clones. And what you can see, again, going back up for the AKT, is that they upregulate phospho-AKT compared to the parental cell population from the same patient. So that pool CD31 that contains both vascular and lymphatic. And you can see as control HDLX and HUVEX, and you can see the AKT is lower activation. So again, these 3 clones for LECs and 3 for VECs were analyzed for the presence of the PIK3CA mutation. And we actually found that the PIK3CA mutation was present in all of the LEC clones and the VEC clones. And in response to this, again, we quantify the phospho-AKT, as you can see here, it's increased. And also these cells have increased proliferation compared to control HDLEC and HUVEC. And this proliferation rate is similar to the parental CLVM endothelial cell population. And in this experiment, what we have done actually, this cell population, clonal population obtained from one single cell were injected in mouse. And what we could see is that the lymphatic clones generated abnormal vascular channels that were only lymphatic. Conversely, the vascular endothelial cell clones were able to form abnormal vascular channels that were only vascular, so where we can see RBCs inside. And we don't see RBCs when we use lymphatic ECs, we see only lymphatic channels. And I want to show you just as control cells, HUVEC and HDLX do not show these vascular abnormal channels. And so in conclusion for this first part, patient-derived clonal LECs and VECs population display constitutive AKT activation, increased proliferation, and can generate lymphatic or vascular lesions in mouse respectively. And in CLVM, PIK3CA mutations are present in both lymphatic and vascular endothelial cells. So what we would like to propose is that the mutation arose probably in a common precursor that was able to then differentiate into the 2 lineages. If you have any question on this part, you can go on now because I'm going to switch gear.
Oh, actually, I have a question, Elisa.
Yep.
Can you hear me?
Yes, I can.
Yeah, this is Brandon Dixon from Georgia Tech. This is super exciting work. I was wondering, have you ever taken your LECs and VECs and cultured them together? And do those with the mutations have an impaired ability to maintain vascular lymphatic separation versus if you did that with cells that don't have the mutation? Does that make sense?
I'm trying to rephrase and see if I understand correctly. So when we have the parental population, the CD31 positive, I didn't show the staining, but they are a mixed population.
So what I guess I mean is, so normally in healthy tissue, right, lymphatics and arterioles maintain separation from each other, and we know that potoplanin and CLEC2 is responsible for this. I'm wondering if part of the phenotype of this disease is lymph blood mixing at the site of the lesion, and I'm wondering if the mutation impairs the ability to maintain that separation. And if you've ever looked at, like, in say one of these in vitro xenograft models, like putting in a clonal that's a mixed, and do they form distinct vasculatures, or is it mixed?
Yes. When we inject a mixture, the parental CD31 population that includes both, oftentimes we see some vessels that have, you know, the lymphatic fluid. We get a sense it's there. You know, I cannot say 100%, but you see this sort of thick, dense material and RBCs. So I do think there is a mixture of lymphatic fluid, even in the xenograft setting, which is, you know, kind of, you know, not 100% physiological things.
Right.
When we do the single clones, we don't see that. Now, the beautiful thing would be to put them back together, maybe, and see if that happens. That's a good idea.
Okay, thank you very much.
Thank you. Okay, if there are no more questions, I'm going to move on to talk about venous malformation. Venous malformations are caused by Tie2 or TEC or PIK3CA mutations. And venous malformations, again, similarly to lymphatic, structurally, you know, there's formation of these massively enlarged networks of veins, which are kind of accessory veins that spread out from a normal. As I talked a little bit about PIK3CA, I'm gonna now spend some time to tell you about what the Tie2 mutations are. And in patients, minority of patients have a germline TEC mutation, but the majority have somatic mutations. They're all activating mutations. And you can see in this diagram, they're located in the tyrosine kinase domain or tyrosine kinase insert domain. They can be found in single or as two mutations. When there are two, they are found in cis in the same allele. And in blue here in this image are the inherited mutations. Now the L914F is highlighted because that's the mutation, that's the most frequent Tie2 mutation found in patients, and that's the one we are focusing our work on. In the Western blot, so then I do want to say that all this beautiful work has been done by the lab of Mika Vikula. And this Western blot on the right side is showing how if you overexpress Tie2, I believe those are fibroblasts, the wild type Tie2 does not induce increased phosphorylation of the receptor, but if you have a mutated form, the R849W is an inherited mutation, you have increased phosphorylation. The L914F, which is a somatic mutation, induces a much higher activation. And here you can see how the combination of two mutations induce sort of an increased compared to each single one. What is Tie2? Tie2 is a receptor primarily found on endothelial cells, and the most well-studied ligands are angiopoietin-1 and angiopoietin-2. Angiopoietin-1 binds to Tie2 and can induce phosphorylation in most cases, and promotes survival of endothelial cells and quiescence. And angiopoietin-1 is primarily produced by the perivascular cells. Angiopoietin-2, again, there are always papers that talk about different settings where things are a little different, but in most general case, shuts off the phosphorylation of the receptor. And in absence of VEGF, there is destabilization of the vessels. But when VEGF is present and communicates with VEGF receptor 2, the two receptors can dimerize and promote active angiogenesis and sprouting. We have generated a xenograft. This, again, this was earlier than the other project, but in a similar fashion. And in this case, instead of using, we've also used patient cells that I will show you later, but we also use normal HUVECs and engineer them to express the mutant Tie2 receptor. After 7 days, the mutant Tie2 HUVECs form these bluish, highly vascularized lesions. And I want to show you the histology. And let's start from the histology of a patient sample. And you can see this enlarged vascular channel that's stained for UEA, which is a marker for the human endothelial cells. And you can see how the perivascular cells that stand for alpha SMA are not really that present in comparison to another vascular anomaly, such as infantile hemangioma. You can see all the small blood vessels have sort of like a necklace, a pearl or necklace of small muscle cells around. And also in our xenograft, you can see enlarged vascular channels, but there are very scarce perivascular cells there. And here you can see in comparison, when you have HUVEC that overexpress the wild type Tie2, we do not see blood vessels, but the endothelial cells just form little clumps there. We've also isolated endothelial cells from these patients, again, from tissue or lesional blood. If you're doing this from lesional blood or venous malformation, my advice is to be patient. The cells don't come out the day after. Sometimes you have to wait for about 10 days. And so I really have to, praise Jillian for all of her patience and work with this. We keep changing medium and waiting and looking at the whole plate to see if something happens. And so it's a work that has a lot of patience involved. But so here is some of the cells we obtained from patients, and we found cells with the Tie2 L14F mutation and cells with the PIK3CA, and also cells with both of the mutations. And here as well, we have found that the mutation is present in the endothelial cells and is absent in the non-endothelial cells. And in terms of signaling, what we see, bear with me with this Western blot. So we have looked at all of these patient-derived endothelial cells, plus minus, or actually minus plus angiopoietin-1, because we wanted to also stimulate the Tie2 receptor and see it, for example, and see if we could augment the signaling. What you can see, phospho-Tie2 is constitutively active in the VMECs with Tie2 mutation, and it's not when you have a PIK3CA mutation. And you are a little bit, yes, when you have both Tie2 and PIK3CA. And you can see here in HUVEC, there is no activation, but when we add angiopoietin-1, you can see it there. And with angiopoietin-1, we can also overstimulate the Tie2 mutant ECs from patients. But also the PIK3CA mutant seem to have an enhanced response to angiopoietin-1 compared to the HUVEC. Next, we look at AKT. And in terms of AKT, you can see that also the Tie2 mutant endothelial cells have a constitutive activation of AKT, but this is even higher in the PIK3CA mutant ECs. And when you have both mutations, this is synergistic, it's increased even more. For what, if we look at the MAP kinase pathway, we look at phospho-ERK, and you can see that this is hyperactive or constitutively active in the Tie2 mutant endothelial cells. And then again, we increase by adding angiopoietin-1, but we don't see much in the PIK3CA cells. And so this shows you that there is a little bit of different signaling there, so that there is more AKT in the PIK3CA mutant cells, and in the Tie2 there is less AKT, but there is more, there is some ERK. And so we don't know, but this could become very important information when designing therapeutic strategies for these patients. We have also generated patient-derived xenografts with these ECs from patients, and here you have PIK3CA mutation and here the Tie2, and it will be evident to your eyes. What I'm summarizing with this schematic is that when you look at VMECs with PIK3CA mutation, we have increased vascular density compared to the VMECs with the Tie2 mutation. Now, this the N number is not very high, but it would be good to add more samples and compare that more extensively. But I think this is what's showing that there is a tendency. And so it's possible that that increased AKT activation in these patients with PIK3CA mutations in VM induces a phenotype that is, correlates with increased vascular density. With the xenograft model for venous malformation, we've also been able to perform studies to look at the effect of different drug treatments And while this now seems kind of a surpassed data, but at the time, this was very important for us that we actually studied in conjunction a type 2 inhibitor and rapamycin, and found that rapamycin was giving us an effect that was much better than type 2 inhibitors. And so we have injected the cells, let the lesion form, and then treated for 21 days. And you can see that the vehicle-treated VM xenografts kept growing, but the rapamycin remained stable. And we found that the effect of rapamycin when given every day was due to a decrease of the phospho-AKT at the site 473. And as you know, there are many clinical trials around the world for the use of rapamycin in different types of vascular anomalies. Let me go back. So because rapamycin was not really promoting regression, was just keeping, I mean, this is still a good result, but we wanted to really see if we could push this curve to for these lesions to actually regress, to become smaller. And we started a phenotypic drug screening that was unbiased, to look for, what we can find in terms of drugs that would be already FDA approved for other indications. And we've tested the efficacy of a panel of drugs, again, FDA approved for other indications, to inhibit cell proliferation. And we have compared to the HUVEC overexpressing the mutant type 2 receptor versus the wild type, and the patient endothelial cells with the same mutation versus HUVEC. And you want to always identify drugs that target your mutant endothelial cells and do not target the wild type counterpart. And so the idea, we wanted to identify a novel drug that could be used alone, or a drug that could be used in combination with rapamycin to promote regression of vascular lesions. Surprisingly, we came across 3 different drugs that were in the same class, such as the ABL kinase inhibitors, nilotinib, dasatinib, and ponatinib. Those are small molecule kinase inhibitors, and so they are dirty, so they inhibit a bunch of other stuff. But the fact that all of 3, were hits in our screen made us look at the intersection between those Venn diagrams. And as you can see, we have here at the top, some factors that are not really expressed in endothelial cells. And then we put our attention on ABL1 and ABL2. PCR-ABL is definitely not the case in venous malformation. There is no translocation. And so what do we know about ABL1 and ABL2? They are no receptor tyrosine protein kinases. They are important for cell growth, survival, cytoskeletal remodeling, and DNA damage response. And their role in endothelial cell biology and vascular development, I would say, is understudied. So in one study, they performed an ABL1 knockdown in endothelial cells in a ABL2 null background. And they reported embryonic lethality due to loss of vasculature. In a second study in the Journal of Experimental Medicine by Christiana Rurberg, they found that the use of ABL kinase inhibitors prevented or showed that there is decreased vascularity in the retina model here. So we wanted to know if c-ABL was constitutively active in the type 2 mutated endothelial cells. And in fact, as you see here, we have HUVEC with non-targeted vector, wild type 2 or mutant type 2, and phospho-c-ABL is exclusively phosphorylated in the mutant cells. And as you can see, for control, there is phospho-type 2. We've also looked in patient-derived endothelial cells. And so here, we've also used HUVEC as control, and we have no angiopoietin-1 and angiopoietin-1. And when we treat with angiopoietin-1, we see that we can stimulate the phosphorylation of c-ABL, suggesting that it's really downstream of the type 2. And in patient-derived endothelial cells, again, with the type 2 mutation, phospho-c-ABL is constitutively active. And then when we add angiopoietin-1, we can furthermore increase this activation. To furthermore explore the role of c-ABL in the formation of venous malformations in mouse. We used the HUVEC expressed in the mutant type 2 and performed the xenograft with cells, the mutant cells with shRNA with knockdown for ABL1 and ABL2. We had to knock down both because when we knock down either one, we have compensation. So to do a genetic experiment that would result in decreased phosphorylation of ABL, we had to knock down both genes. And you can see that compared to the shRNA scramble, the lesions are smaller and they are less vascularized, as you can see all in the quantification for the percentage vascular area. So all of these together suggest that ABL has an important role in the formation of venous malformation. We chose, I don't have all the data how we came to choose ponatinib, but it was compared to the other ABL inhibitors, this was the strongest and the one that we could use at a smaller concentration. And we performed experiments in mouse, again in the xenograft. We injected the cells, the mutant cells, we let the lesion form and then treated. And in this waterfall plot diagram, you can see that the size of each lesion that we measure keeps increasing in response to vehicle treatment. But when you treat with ponatinib or rapamycin, there is still probably a little bit of increase, but it's minimal. And the beauty of this is when we combine ponatinib and rapamycin, we can promote a regression of these lesions. And we can also decrease the dose of each single individual drug and still see a decrease in size. And also with this other type of graph, when we look at the lesion size longitudinally, you can see that we can push a regression in size, but there is no rebound. We've also done the same thing with patient-derived endothelial cells with the same type 2 mutation. We have 2 patients here, and you can see ponatinib, rapamycin, there is decreased vascularity, and when you use the combination, there is even less vascularity, as you can see also from the staining of Ulex europaeus agglutinin 1, again to stain the human blood vessels in the xenograft, and here it's the quantification. What is the drug combination doing? We have looked at proliferation with Ki67 and at apoptosis with the CLVM marker. And what happens is that proliferation is decreased in combination or reduced dose combination. And CLVM, the apoptosis is increased. Again, it's quantified, decreased proliferation, increased apoptosis. We were interested in understanding what would happen when we actually take off the treatment. And what you can see here is that at the moment when we stop at day 28, there is lesion rebound. And so we hypothesized that by adding rapamycin cream to lesions that were already decreased in size, we could keep the lesions small. And this is what in fact happened. You can see that the lesions stayed small compared to the vehicle cream. And here you can see the difference in size. When the vehicle, they kept growing from day 28 on when we stopped the drug, and the rapamycin cream stayed stable. If not, they kept actually shrinking. In conclusion of this part, I've shown you that the type 2 mutation, the L14F, at least, that we focused on, promotes activation of the type 2 receptor, the AKT, phospho-c-ABL, and ERK. And if we leave these lesions untreated, they will keep growing. If we treat with single drug, they remain stable, rapamycin or ponatinib. And when we combine, we can promote ponatinib with rapamycin, we can promote regression. And when we remove the systemic drug combination, and if we keep with topical rapamycin, we can let these lesions stay small. I have few more slides, and I want to use that to tell you a little bit about a system that we've been using to study venous malformation, but also other lymphatic diseases in collaboration with Tim Lacrasse. And this system we have sort of developed in conjunction with work done by George Davis, and we really obtained preliminary data really thanks to his input. And I really want to show this in case anybody wants to, you know, start to model vascular anomalies, and if you have any question, anything you want to ask. And the beauty of this system is that, as I will show you, is how we can model lumen formation, and we can also use this to test therapeutic drugs. The way this works is to plate endothelial cells, let them grow, and then seed them on top of dextran coated microcarrier beads, and then embed this sphere of endothelial cells into a fibrin gel. And we put fibroblasts on top here. And the fibroblasts really are responsible for secreting a lot of factors that stimulate luminogenesis. And in these images here, you can see that HUVEC can sprout and form these lumens that are normal, longitudinal, and you can see in the 3D rendering, so the sort of half cups are the beads, and that there is a lumen here. When we use the mutant type 2 cells, initially we just saw like a sheet of cells everywhere, but then the 3D rendering helped us to understand that what we were seeing was just the roof of these massively enlarged structures that had a hollow inside, as you can also see in this. Okay, maybe I cannot show you.
I think because you have your laser.
Oh, yeah.
You switch to your pointer.
Thank you. And so then it's going to switch to a 3D rendering view where it's easier to see the hollow inside. So with this system, we've been able also to add parasites. And when you use HUVECs, so we've used a HUVEC labeled with GFP and parasites with blue fluorescent protein. And you can see that they can really organize where the endothelial cells form these lumens and the perivascular cells really wrap around, so that if you look only at the parasite image, it mirrors sort of, you know, what you see with the HUVEC, so that they really sit on top of these lumens. But when we use the mutant type 2 endothelial cells that I've shown you before in patients, there is a lack of perivascular cell recruitment. In this model, we see the same thing. You can see here this enlarged vascular structure. In the one field view, you can see that it's empty inside, and the perivascular cells are just sitting around there, but not preferentially sitting to wrap the vascular structure. And you can see that when we quantify the parasites per vascular area, they are that number is decreased. And this is not due to an effect of decreased proliferation induced by the mutant cells. The last data slide that I will show you today is to show you how this has been effectively used to test the effect of different drugs. And so what we can do here, we can start the system and let the endothelial cells form lumen, and then start a drug treatment and see if you have regression. And as you can see here, this is a 2D image, so it's a little bit harder to visualize the 3D stuff, but we have a microscope where we can take images of the same exact spot on the same at different time points. And so we can really follow how this structure progresses from day 8 to 14. And you can see in vehicle, these lesions keep growing. Ponatinib or rapamycin alone, there is still a little grow, although much reduced compared to vehicle. And then when we have the drug combination, there is regression of these vascular lesions, as seen in this quantification. And with this, I am going to conclude, and I'm going to thank members of my lab for this all this work, and all the collaborators. Importantly, we've done a lot of beautiful work in collaboration with Team Lacrosse, and also all the clinical collaborators at Cincinnati Children's, the patients and families, and also that sort of interdisciplinary work that we do all together with the clinicians, patient support groups, and scientists. And last of all, I'm going to thank all the funding sources. And thank you so much for being here today.
That was a great presentation. Very beautiful work. I'll get the discussion started, and then we have some questions in the chat that I'll read. But I was wondering if you could speculate as to the mechanism by which Tie2 leads to the activation of ABL.
I think it's pretty much direct because we have data that the two of them bind together. We have done co-IP and they bind together. We haven't been able to publish that stuff, but we've seen that the two bind together.
Great.
And I think also when you inhibit c-ABL, then we start to see decreased. So there's like a feedback loop. I think there is a very finely tuned crosstalk between the two of them.
And maybe I'll sneak in another question. You've shown in your in vitro experiments that the addition of angiopoietin-1 can further enhance Tie2 signaling. Is angiopoietin-1, do you think it also plays a role in vivo, that maybe it's in the microenvironment and could be further promoting Tie2 signaling? Yeah,
I've been thinking about that because there are less perivascular cells. So either there's something else around there, fibroblasts, and something that contributes to that. But because there are no or less perivascular cells, I wouldn't know exactly where that would come from. Yeah, it's an interesting speculation. Yeah.
All right, Phil King has a question, and I'll just read it. Unless, Phil, would you like to unmute yourself and share your video?
Yes, it's a question that you might not have a ready answer to, but I think it's one that's important, which is, why is it that through the study of these different vascular anomalies, that it appears that gain-of-function mutations in the PI3 kinase signaling pathway result in venous malformations and lymphatic malformations, whereas gain-of-function mutations in the RAS signaling pathway result in lymphatic abnormalities for sure, but peculiarly arteriovenous malformations or capillary malformations. Do you have any thoughts upon why there would be that distinction?
I mean, PIK3CA mutations have been found in both venous and lymphatic. So I know in the lymphatic side of the coin, there are also RAS mutations. So, and I think, I just don't know. I think that RAS mutations are probably more deleterious. That MAP kinase signaling promotes more proliferation than the PIK3CA. But I honestly, I don't have an answer.
No, there is a distinction, right? So you don't see gain-of-function PI3 kinase mutations, as far as I'm aware, in arteriovenous malformation.
Yes, in that setting, yes.
And unless somebody in this forum corrects me, I'm not sure you see gain-of-function mutations in the MAP kinase signaling pathway in venous malformations.
Well, but we do see that there is the Tie2 promotes both AKT and also ERK activation. So, well, we haven't ruled out that the ERK part is really necessary for the
right. All I mean is that there's no evidence of mutations within RAS MAP kinase signaling pathway components themselves.
Yeah, so I can't fully answer on the arterial part because we're not doing much work on it, but there is a bioarchive, a preprint from Mariona Graupera, where they've looked in the retina with this PIK3CA mutation. They put it, I believe it was a V cadherin promoter, and they show that these vascular malformations form only in the capillary and venous plexus, and they are never found in the artery.
Yeah.
So there might be a delicate balance where if that mutation happens in an artery, I don't know, that's some protective mechanism.
Okay, thank you.
Thank you. It's an interesting question.
All right, our next is Catherine. Which, actually, is that Brandon?
I think so.
Sorry, I guess my daughter signed into my Zoom account on my iPad. She won't be asking the question, I will. Thanks again. I always enjoy it. We've been really inspired by your work. Maybe what I'm asking you here to do is to help me write my own grants. But when I think about 3D models in sort of that they're varying complexity that they can have and what they really help us with. Do you think if we do a screen in a 3D model, a drug screen, we're going to learn anything that we didn't learn in just a simple, because right, I can do 2D monolayers and put these on plates and do high throughput and test tons of drugs. And it's much more complicated to do that in 3D. Do we need the 3D model to do that? Do you think the 3D model is going to give us a hit that was negative in the 2D model, or vice versa? We have a hit in the 2D model that turns out not to work. Like, where do you see 3D models fitting in sort of this?
I like them in the sense of looking for regression, which is a specific phenotype where those things are already formed, because you can always prevent proliferation, but really to show that in a preformed, malformed vascular channel, you can act there and induce regression. I think that's a very powerful system.
Okay.
So,
yeah, that's cool. It would be cool to see if you could show like you can't capture that regression at all in like a 2D context, and you really need. But thanks. That's a helpful idea. I'll be using that rationale as well. Thank you.
But then you can look for other things like the perivascular coverage. Like if you can use the system to add more matrix specific or other contributors of disease, so that then that's. That's more powerful.
Thanks.
All right. Fayez, if you want to show your video and ask your question.
Sure. Thank you for great work again. Fayez Safari here from Neomid, Ohio. I have a question regarding those xenograft. I mean, do these lymphatic vessels spread to, if you leave them longer, do you think they're going to spread to other tissues, or they stay within?
We haven't looked that long. For venous malformations, we have done also up to 4 or 6 weeks, and we haven't seen any spread.
An invasion to other stuff. So the other question I have about c-ABL, I mean, there are enough evidence about this protein when it comes to skeletogenesis and bone cells differentiation. Do you have any suggestion that the mutation in relation to the c-ABL protein has anything to do with probably the invasion onto the skeleton?
Invasion, like what we see is defects in the cytoskeleton rearrangement. So we think that's what's happening. And in fact, these cells, upon introduction of the mutation, their cell shape changes, they become more spindle. And we are trying to look at also a little bit of that.
Gotcha.
But I don't have enough
Thank you
to present. Yeah.
Thank you. That's great work.
Thank you.
All right, Håkan Arsen, if you want to ask your question.
Thank you, Mike. Great talk, as always. You did some combination therapies in the mouse model with the lesions that you had, and sort of, given what's known about the sort of the escape mechanism in the signaling pathways between the PIK3CA and RAS-MAP kinase, do you have any sort of a clear evidence or data that combination therapy in human is beneficial? That combination with sirolimus with something else, or palliative with something else is superior or above an individual therapy? Is there any such data?
Yeah, I mean, in our own work, I've shown you how the combination was synergistic. I haven't shown all the data about the synergism, but that's what's happening. What I would say is that when we were looking at rapamycin, what we would always notice is that while AKT was going down, we would inhibit AKT activation, ERK at the same time goes up, so that there is a compensation. So the cells are always going to try to find their own way to survive when you give them an inhibitor. And the endothelial cells really rely on these two major pathways, the PIK3CA and the MAP kinase. And so I do believe that combination will always be better. And maybe you can, bring down the dose of each component and limit toxicity. But then, of course, sometimes when you combine the two drugs, you can have additional side effects. So one has to look at this carefully. But we have seen that with the use of single drug, while you affect one pathway, the cells try to compensate. Does that answer your question?
Thank you. Yeah. Thanks.
Thanks.
All right. Well, if there are no additional questions, I just want to thank you, Elisa. This was a great presentation. Thank you, everyone, for attending. It was great to see a lot of you over at ISFA in person. And we'll see you next month for our next seminar. So thank you, Elisa, once again.
Thank you. Thanks again.